Hello to all of you. I am the president of the French Society for Cell and Gene Therapy, and the board and myself are very happy to present to you this webinar on gene therapy and hemophilia. We have already invited several experts to give webinars on different subjects, with our last series treating about gene editing for gene therapy. And these webinars are still available on our SFTCG webpage and YouTube channel. So I really invite you to have a look at them. But the current webinar will be presented by Dr. Thierry Vandendriessche. He is a tenured full professor at the Vrije Universiteit Brussels in Belgium, the Faculty of Medicine and Pharmacy, where he is founding director of the Department of Gene Therapy and Regenerative Medicine. He obtained his PhD in Brussels at the VUB in 1992 in the field of gene therapy already, and was a visiting fellow at the Weizmann Institute for Science in Israel. He continued then his research in gene therapy as a postdoctoral fellow at the National Institute of Health in the States, in the labs of Dr. Rick Morgan, Dr. Mike Blaise, and Dr. French Anderson. And later he worked with Dr. Desiree Collen at the University of Leuven in Belgium. He previously also was the president of the European Society of Gene and Cell Therapy, and he was the recipient of the ESCGT Outstanding Achievement Award. And we are really very happy to have invited you for this webinar, Thierry. And I please you can start with your talk. Thank you.
Thank you very much, Dr. Verhooyen. Thank you, Els, for this very kind introduction. It's, of course, always a pleasure and an honor for me to contribute to the activities of the SFTCG. I'm really very happy to be here once again on this virtual platform. And please feel free to our audience to send us questions during this talk using this live chat window. You need to sign in to YouTube with either YouTube or a Google account. These live chat messages are not private, so everyone who tunes in can read them. And if you're not able to reach us through these live chats, feel free to send your questions through this email, info@sftcg.fr. And this seminar was made possible with the support of Pfizer Maladies Rares. So without further ado, I'd like to start off. And I know that France this week, this upcoming week will be the center of the earth, so to speak, if not the universe, with the Tour de France and Olympics all coalescing in France. But perhaps you will find a little gap in your schedule to actually watch this seminar on gene therapy for hemophilia, the end of the beginning. The complete human genome was sequenced in the year 2000, resulting in a significant scientific milestone, to the extent that the then president of the United States, Bill Clinton, invited some of the main stakeholders in this endeavor, Craig Venter, representing the private sector, and Francis Collins, then director of NIH, representing the public sector, to a celebration in the White House. And I just wanted to quote President Clinton, who then said, "With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases." Granted, it was a bold statement, but I think it's very important to be bold as a politician, and certainly when it comes to driving the scientific endeavor forward. Now, what really inspires me to start my gene therapy career for the last 40 years is the fact that a single point mutation, a single error in the genome, the human genome of 3 billion nucleotides, can make all the difference between life and death. it can result in a potentially life-threatening disorder like Duchenne muscular dystrophy, cystic fibrosis, and of course also hemophilia, which is the subject of today's presentation. And this is equivalent to one typing error in 15 entire volumes of the Encyclopedia Britannica. So imagine you have to type out every single page of every volume 15 times and you make one typo. Well, that's what gets you into trouble if that happens at the genetic level in our genome. Now, luckily, we have moved a long way now since the early days of gene therapy. And there are a multitude of gene therapy products that have now been approved by the European Medicines Agency and by the United States Food and Drug Administration. I've just listed them here, and they cover essentially all of the different systems and organs and a whole plethora of diseases affecting the central nervous system, the liver, the immune system, you name it. And typically, two types of gene delivery vehicles were being used to achieve these therapeutic successes. On the one hand, lentiviral or retroviral vectors, mainly in the context of ex vivo gene therapy. So one has to take out the cells out of the patient, engineer them, and put them back. But also in vivo gene therapy has been pretty much moving forward thanks to the development of adeno-associated viral vector-based gene therapy. Now, just to say a few words about hemophilia, it's caused by a defect in clotting factor gene 8, causing hemophilia A, or clotting factor gene 9, causing hemophilia B. And whether it's hemophilia A or hemophilia B, the symptoms are very similar. We're getting uncontrolled bleeds in soft tissues, in the joints primarily, resulting in severe arthropathy, as you can see here on the slides. And sometimes this can be extremely painful, resulting in a crippling consequence. Now, initially, you know that hemophilia had an important historical significance because Tsarevich Alexis was actually suffering from hemophilia B, and you can see him posing here for the picture, and you can notice that he's not keeping his legs straight, and the reason for this is that he had actually joint pain, and this sort of alleviated some of the discomfort that he felt. Now, some individuals in the court of the Tsar took advantage of this medical condition of Alexis to gain more and more influence in the Russian court. No one less than Rasputin himself. And I would say that anything that rhymes with Putin should be looked at rather skeptical, isn't it? And yes, Rasputin, of course, just like the Tsar family at the end of the day in 1917 got executed. Now, typically, enough of this historical background, but typically nowadays when we want to treat hemophilia, we administer either plasma-derived or recombinant clotting factors. And recombinant clotting factors that are being administered prophylactically at a regular time interval, several times per week, is really now the standard of care. And that's great. You can achieve some level of protection, but of course, these clotting factors, they are not stable. You reach very high peaks and then the levels drop down again. You have to re-inject, the levels go up, the level goes down. So that's not an ideal situation because this results in a transient correction of the hemostasis. So ideally with gene therapy, we aim to obtain sustained expression in the therapeutic range that would then stably correct the hemostatic defect. Now, what is a therapeutic level? Well, strictly spoken, it's more than 1% because any level below 1% is associated with severe hemophilia. But ideally, we're thinking of levels 30, 50%, there we would already have a very significant effect indeed. Now, the Achilles heel in the field is undoubtedly the vector itself that we need to deliver the therapeutic cargo, in our case, the factor 8 or factor 9 cDNA, into the target cells of choice. And typically, when we think of in vivo gene therapy, there's a very narrow therapeutic window. And this is actually also very much independent of the disease, because we've been seeing this in many types of indications, meaning that the therapeutic effects increases with increasing vector dose. There's typically a dose response, but so do the toxic effects. So at a given vector dose, toxicity emerges, and the difference between the therapeutic efficacy and the toxic effects defines the therapeutic window. And it's actually fairly narrow indeed. And what we are trying to achieve in the field is to broaden this therapeutic window to the extent that we can maximize the therapeutic efficacy while still having only a minimal toxic effect. That's really one of the holy grails, if you may, in the field. And there's various ways of trying to achieve this. So everything boils down to the vector itself. This is the essential tool that we need, but it's also at the same token the Achilles heel of our field of gene therapy. So how can we improve the overall efficacy? How can we minimize all these safety concerns? How can we broaden this therapeutic window? Well, we can bet on 3 horses at the same time. We can try to improve the vector itself. We can try to modify the promoter that is used to drive the expression of the gene of interest. And last but not least, we can try to tamper with the gene sequence itself, either altering the expression levels, but perhaps more interestingly, altering the activity of the gene of interest. And in the context of hemophilia gene therapy, I will try to illustrate this molecular triad concepts, how we can play with each of these important confounding variables to maximize efficacy, minimize toxicity, and improve the therapeutic window. So the ultimate goal really is to use an as low vector dose as possible, because we know that above a certain threshold, we end up with toxicity, typically liver toxicity, when we use AAV, that is. But below a certain threshold dose, we hope to avoid this altogether. But of course, if your dose is too low, you may pay a price in terms of efficacy, because you may not reach the therapeutic levels that are required to achieve a significant biological and therapeutic effect. So it's very important to make sure that your vector still remains sufficiently potent. So let me just illustrate this concept of these molecular triads, beginning with the promoter that we use to drive expression of the gene. So we've actually adopted a very unbiased approach rather than a trial and error approach where we just trial and compare different liver-specific promoters. We use the genome-wide computational analysis of promoters of genes that are highly expressed and exclusively expressed in the liver in order to identify certain motifs in the human genome that are associated with high liver-specific expression. And we figure once we identify such motifs, perhaps we could use them in conjunction with the gene therapy vector to maximize expression of the gene of interest. So to make this a little bit more specific, these DNA sequences correspond typically to transcription factor binding sites that are typically located upstream of the promoter, although they could also be internal or even downstream of the gene sequence. Now, they also evolutionary conserved. That's an additional filtering that we implemented from the start, because there's no point identifying an element that is highly potent, let's say in mice, if it doesn't translate to human. But luckily, we started off with human DNA to begin with, and we specifically focus on evolutionary conserved sequences in the hope that if we do see an effect in a mouse or in a dog or in a monkey, that it will eventually translate to human. So typically, this is like an extra feature on the engine that we need to go forward. So this is like a molecular turbocharger that is used as an expression booster. And one of these elements that we identified were actually fairly small, surprisingly. You would think you would need a large element to achieve a significant effect, but we found elements of only 72 bases in this case that bound a few quintessential liver transcription factors like NHF1, along with other factors like CBP and FOX. And altogether, these factors cooperate to actually maximize liver-specific expression. In fact, we made an AAV GFP vector prep, 10 to the 12th vectors, the entire vector prep. We injected into a single animal to see if our expression was specific for the liver. And as you can see here, the minute you have a GFP signal, the liver cells turn green, but we didn't see any green cells in any of the other tissues that we tested. The blue refers to the DAPI nuclear staining. So clearly, even at high vector doses, the promoter that we use in conjunction with these molecular turbochargers were fairly specific for hepatocytes. Now, I'm not saying that there is absolutely no expression in any of these other tissues, because we do know that the expression in other tissues is also depending on the vector capsid, as I will come back to later. Now, how does this fit into the hemophilia gene therapy story? Well, we plugged these cis-regulatory modules, or CRMs, as molecular turbochargers, if you may, upstream potent hepatocyte-specific promoters. And one of the most potent ones is the transthyretin promoter. It's also a relatively small promoter, yet powerful. And we could see that the incorporation of such an element resulted in a 5 to 10-fold increase in factor IX expression when driven from the TTR promoter. And this was quite significant because we could attain physiologic levels of factor IX in hemophilic B mice at fairly low doses, 5 × 10⁹ genome copies. And these levels were stable. We've actually moved this all the way to non-human primate studies and obtained levels approaching 40%. This was also done in collaboration with Federico Mingozzi and Omeya Ajani in Nantes, and we published this work. Now, the good news is that we are attaining 40%. And if you remember what I said a few minutes ago, if we can end up between 30 and 50%, that would be great, because if a patient has 30 to 50%, even though it's not normal yet, it would already be a significant improvement of the patient's quality of life. Now, this is of course great, but we all have to be a little bit wary when it comes to transcriptional control elements. As we know from the experience with lenti and retrovectors, sometimes these elements carry a greater risk of insertional oncogenesis when they activate neighboring genes upon integration of a vector in the genome. So to actually assess that risk, we plugged in these turbochargers, this CRM, with a liver promoter, the same one as we used in AAV, expressing factor IX, different types of factor IX transgenes. And we used a piggyback transposon, a hyperactive platform, to deliver this construct by hydrodynamic delivery in hemophilic animals. And we also tested it in tumor-prone mouse models that are super sensitive to the formation of hepatocellular carcinoma. These are DEN-induced, so we use a carcinogen to induce cancer. And the minute you have let's say, a cooperative effect from whatever you co-administer to the mouse, you should see an increase in hepatocellular carcinoma. Now, the good news is that we didn't see that. So if you look here at the number of nodules in this super sensitive hepatocellular carcinoma model, we have here the control. This is just DEN. Yes, we do see some nodules here. Even if we just inject PBS in this mouse by hydrodynamic transfection, we get a certain local inflammation, and we do see a small yet significant increase in the number of nodules. This is just an illustration how sensitive this model really is. But then, if we express factor 9 with these turbochargers, if we really have an increased risk of insertional oncogenesis, we should see an increase in nodules, and this doesn't happen. So this gives us some reassurance that these turbochargers, even though they can result in a potent increase in activity, in expression levels, they do not increase the risk of insertional oncogenesis, since we don't see an increased risk of hepatocellular carcinoma incidence in this experimental model. And I haven't come across any other model that would be more sensitive than this one. Typically also we look at survival. We have a number of controls with a c-MYC delivered as a transpose, and of course in that case all the animals die of liver cancer, but in this case none of the animals develop liver cancer in the first place. So efficacy, yes. Safety, yes. So I think we're pretty reassured about this. Now let me shift gears and talk a little bit more about other strategies we can employ to maximize efficacy and minimize toxicity. And I'm shifting gears now from the promoter to the gene itself. And many of us are using codon optimization to maximize the expression level. And this is something we and many others have been doing, but we also try to find ways to increase the activity. How do we do that? Well, we can learn from nature and from other clinical experiences, because a few years ago, my friends and the late Valder Arruda published a paper along with Paolo Simeoni of an X-linked thrombophilia patient who carried a mutation, a point mutation in the factor 9 gene. The patient was from Padua, and hence the mutation was then coined as factor 9 Padua, a beautiful Renaissance city in Italy. And this actually literally triggered a renaissance in gene therapy for hemophilia B, because we thought, well, that's great, even though it causes thrombophilia in these patients, too much clotting, so it's a problem. But we thought that in the context of hemophilia, this is totally different. Maybe we should incorporate the factor 9 Padua transgene in a vector in order to maximize the activity. So typically we took our turbocharged promoters expressing the factor 9 Padua and then administered these vectors, they were AAV9 by the way, to a hemophilic mouse. And we could see a very significant increase in activity, 5 to 10-fold, as shown here in blue, compared to the red circles here, which show us the antigen levels, the protein level. The activity is shown in blue. So rather than having a 1-to-1 relationship, we have a 5 or even 10-to-1 relationship of activity versus antigen. So that can eventually trigger an important dose advantage if it allows us to lower the vector doses while still obtaining a significant therapeutic effect. And as I said, this triggered a renaissance in hemophilia B gene therapy and actually resulted in very encouraging clinical studies where an AAV5 vector was used, and these studies were conducted by AMT and Unicure, resulting in a mean factor 9 activity of about 30 to 40% in patients that had either no neutralizing antibodies to AAV, and even in patients that had some neutralizing antibodies to AAV, even though low levels, they could detect up to 30% factor 9 activity. So that was really great. Of course, if the neutralizing antibody titer against AAV is so high that it immediately neutralizes any trace amount of AAV, it's not going to work, of course. So, but below a certain threshold, it's possible to still achieve sustained therapeutic effects, even in the face of neutralizing antibodies to AAV. That's just a little side note here, but really what I want to emphasize is these very high activity levels of factor IX. There were some safety issues still, transaminitis in some of the patients that required corticosteroid treatments. Important here is that there was a significant drop in annualized bleeding rates from 4 bleeds to 1.5 bleeds in the post-treatment periods, implying that, yes, indeed, we can drastically reduce it, but not completely prevent it. So there's still some room for improvement here. And I will come back to that later, how we could further improve beyond Padua. Now, nevertheless, it's clear that this was a major step forward and resulted in the first hemophilia B gene therapy product that was approved. That was more than 2 decades in the making, in fact. And also more recently, the US FDA approved Pfizer's Beqvez product, which is also based on the factor IX Padua. Now, can we move beyond Padua? Well, I think we can, because we can incorporate additional mutations in the factor IX transgene to further maximize activity. And we collaborated with Catalyst Biosciences, with Grant Blaus and colleagues, to actually test that. They had engineered a factor IX transgene that contains 3 mutations at 3 different locations, resulting in increased catalytic activity of factor IX, increased resistance to antithrombin inhibition. So in a way, you prevent the brake from working, and to improve the affinity for activated factor VIII. And we saw, at least at the protein level, it certainly results in improved clotting efficacy. And we plugged it into an AAV vector to actually check how this works in hemophilia B mice, using factor IX Padua as our gold standard, as our benchmark. So in blue here, you can see the Padua. This is what's now all been approved for use in humans. And here we have the CB variant with the 3 different mutations. This is a specific activity. In red is the CB, in blue is the Padua. We can see a significant increase in activity at different time points with the CB version. We can actually also measure the bleeding time. And again, here, consistent with the increase in specific activity, there is a significant shortening of bleeding time with the CB variant. The full name is CB2679DGT, but let's just call it CB. So this is again moving beyond Padua, giving us a handle to lower the vector dose even further, or perhaps even if we don't want to lower the dose, remember that there are still some patients that show bleeds. If we can use this type of variants, it would also minimize the risk of bleeds, even less so than in the case of Padua. And if you're interested, this work was published by us. Now, what about factor VIII? Well, factor VIII is a whole different story. It's much more challenging to express high levels of factor VIII. There are bottlenecks at every single level, at the level of transcription, at the level of translation, because once the factor VIII protein is formed, it gets kind of clogs up in the endoplasmic reticulum. It is retained there because of the BIP protein that binds factor VIII. And then it's only the beginning of the journey, because factor VIII still then has to traffic to the Golgi apparatus before it's eventually secreted extracellularly. And even during that last trajectory, the mannose residues on the factor VIII glycoprotein can then bind to another protein, LMAN1, that also impedes trafficking. So as you can see here, many different bottlenecks on the way. It's actually starting to look like the ring around Paris or Brussels, for that matter, a lot of molecular congestion. So how can we relieve that congestion? Well, there's a way around it, luckily. Well, first of all, if you want to boost the expression, you can use stronger promoters, which is what we have done with the CRM turbocharged promoters that we use to express factor 9 and factor 8 that also works for factor 8. We can codon optimize factor 8 to improve translational efficiency. We can reduce BIP binding. So BIP is sort of a chaperone, and we can introduce a point mutation in factor 8 so that factor 8 is detached from its chaperone. It doesn't interact so well, and therefore it can traffic better throughout the intracellular trafficking pathways. We can even alter the glycosylations so that, again, also here at the ER-Golgi transition, things go far more efficiently. Now, the good news is that B-domain deleted factor 8 cDNAs have now been incorporated in AAV. There's not much space left because the factor 8 cDNA is quite large, 4.3 kb to its shortest form. Doesn't give you much leverage in terms of promoters and regulatory elements and so forth. But the good news is that you can actually still achieve functional expression of factor 8 in animal models, but also in humans. At the end of the day, an AAV5-based trial was conducted, resulting in therapeutic levels of factor 8. And here you see the factor 8 activity over time. This was over 2 years. And what is immediately obvious is that we are hitting definitely therapeutic levels, but that these levels eventually taper off somewhat, even though they, at this point in time, have stabilized. Most importantly, the bleeding episodes have drastically been reduced after gene therapy, and it doesn't even require any additional factor 8 infusions, at least for a certain period of time. So that's actually good news indeed, and resulted in the first conditional marketing authorization approval for hemophilia A gene therapy in the EU. In the meantime, this has also been approved in other jurisdictions as well. So yes, this is one step in the right direction, even though there are still some issues about the stability of expression, which need to be sorted out. So in a nutshell, what have we learned after all these years? Well, we can achieve therapeutic factor 8 levels, 20% at least at 5 years. Therapeutic factor 9 activity, 30 to 40% at 1 year. And this is conservative. It's actually 30 to 40% for many more years, but the latest reports have not been published in peer-reviewed journals. There's a significant reduction in annual bleeding rates, a discontinuation of prophylaxis. So the patient is off the hook, and that's really important. They don't need to be treated with factors prophylactically, at least many of them, not all of them. It's reassuring that none of the patients develop neutralizing antibodies to factor 8 or factor 9. They do develop antibodies to AAV, of course, preventing readministration, but they don't develop antibodies to factor 8 or factor 9. And this is reassuring because it's known that in some patients that when they receive recombinant factor 8 or recombinant factor 9, that some of these patients do develop neutralizing antibodies to the clotting factor, which are coined as inhibitors. Of course, there's some bias here because these patients have already been pre-screened. They don't have any active inhibitor developments. And I think that is therefore not surprising that they didn't develop inhibitors. I don't think they even had a history of inhibitors to begin with. It turns out that factor 9 is also more stable than factor 8. We don't fully understand why. There's some data from biopsies of the patients that suggests that the transgene may be repressed in patients, accounting for the decline that was seen. There are other issues that we don't fully understand. The patient-to-patient variation, which is quite significant. It's still very much like Russian roulette, if you may. You can have 10% factor 9, or you can have 100% factor 9. And it's not always easy to predict where you will land as a patient. And there's some issues of liver toxicity. Although this is manageable to some extent by corticosteroid treatments and other modalities. But Again, the toxicities that we observe in the hemophilia studies are not as significant as what has been reported for other disease targets, for which much higher vector doses were required. We're talking about more than 10 to the 14th vector genomes per kilogram, as in the case of Duchenne, SMA, or myotubular myopathy. In that case, there were toxicities beyond liver toxicity, resulting in other severe adverse events, acute kidney injury, complement activation, hemolysis, thrombocytopenia, GI bleeds, hyperbilirubinemia, and sepsis. But we didn't see that in any of the studies here for hemophilia, probably because the doses are still below this potentially dangerous threshold. So, to paraphrase Winston Churchill, this is not the end. It's not even the beginning of the end, but it's perhaps the end of the beginning. And there's still some unanswered questions and unsolved problems, but I think we can try to tackle those in very much the same way as we have tackled the challenges that we were faced with 15, 20 years ago. One of the challenges pertains to this interpatient variability. Yeah? Why is it that some patients have very high levels of factor VIII or factor IX and gene therapy, and other patients have relatively modest levels? And again, this is not unique to hemophilia gene therapy. We've seen that in other trials as well with other transgenes. And we can only speculate at this point, and there is some circumstantial evidence from the literature, from preclinical studies that suggest that this may play a role. There could be differences at the level of AAV transduction. Of course, the more efficient the transduction, the higher the chance that you will end up with higher levels of expression. Maybe there's differences at the level of receptor or co-receptor expression, differences at the level of intracellular AAV trafficking, but perhaps also differences at the level of the expression itself, at the mRNA level, translation, secretion, and trafficking. Maybe even to some extent pharmacokinetic and pharmacodynamic differences in the protein levels. We know that from protein substitution therapy that this can be an important variable. And last but not least, differences in immune responses, perhaps differences in innate immune responses. And there's some evidence obtained by Fritz Schäflinger and others that interleukin-6 polymorphisms may play a role in this. So, as you can immediately appreciate, this black box is still very black indeed. We only have a peek into this black box, into some of these potential explanations, but we need some additional hard data to pin this down. Now, as I mentioned in the hemophilia A gene therapy trial, there is a tendency for the expression to taper down. Eventually, it does seem to stabilize after 5 years. But the million-dollar question, and perhaps we should take that literally given the costs of hemophilia A gene therapy, is whether or not the expression will be lifelong. And that's really the holy grail. Will it be lifelong? Does a single shot, is that really a one and done treatment? Well, in mice, yes, for sure. We have cured hundreds of mice, so do many other people. Dogs have also been cured. It's not a problem, but in humans, we're not quite so sure yet because the experience is much more limited in time. And one of the issues that may have an important impact on the expression duration is the genomic integration of the transgene product. So to integrate or not to integrate, to paraphrase Shakespeare, that's the question. Because what happens if your factor VIII or factor IX transgene doesn't integrate? We know that hepatocytes slowly turn over in adults. And over decades, perhaps this would result in a gradual loss of non-integrated factor VIII and factor IX transgenes. As we know, the AAV episomes, they hang around in the nucleus. They're relatively stable, all right? But relatively stable is no guarantee for lifelong stability. So even in the face of slow hepatocyte turnover, perhaps the expression will eventually taper down. So whereas if your transgene integrates stably in the target cell chromosomes, this is a one-way ticket. once you go in, you cannot go out anymore when we use an integrating vector. And in that case, even in the face of rapid hepatocyte proliferation, the expression is believed to be sustained. And actually, we tested that many years ago. I will come back to that in a second. And this has important implications, of course, for pediatric hemophilia, where pediatric subjects have rapidly dividing hepatocytes. And if you were to use a non-integrating vector like AAV in a young child, the expression is believed to be transient. Now, we tested this hypothesis. If you were to use an integrating vector in newborns with rapidly dividing livers, what would then happen? Well, we did see sustained expression of factor 8 in that case when we use an integrating vector. In this case, it was a gamma retroviral vector that we injected in newborn hemophilia A mice. So it was an injection when these animals were 1 to 2 days old. That's it. Just 1 or 2 shots. And then we waited until they died of old age and they expressed factor 8 for their entire life. So a single treatment resulting in lifelong expression. So that is truly a one and done paradigm. It also resulted in immune tolerance to factor 8 in at least half of the animals. We've confirmed this also with lenti, not just in newborns, but also in adult mice. We could see sustained expression with factor 9 when we use lenti. We also use our Padua factor 9 here, resulting in superior activity, just like what we showed with AAV in collaboration with Luigi Naldini and Alessio Cantori. This was actually one of the first preclinical validations of factor 9 Padua in the context of gene therapy. Now, lenti is great. As we know, we could result in lifelong expression. It's been used successfully in the context of ex vivo gene therapy. But what about in vivo gene therapy? Could we use lenti for hemophilia A or B gene therapy? And the results that I just showed suggest that this may indeed be very promising, even in the face of slow or rapid hepatocyte proliferation. Now, but lentis are not perfect. They do transduce hepatocytes, and that's great, but they also go to antigen-presenting cells like macrophages and Kupffer cells. And we published this more than 2 decades ago with GFP reporter genes. We have really significant hepatocyte transduction, but also significant expression and transduction in antigen-presenting cells like splenic macrophages and Kupffer cells in the liver. Now, there's a way around that, and the group of Naldini actually modified the envelope. Instead of using VSVG, they actually incorporated molecular don't eat me signals into the lenti particles, CD47, to block entry of the vector into antigen-presenting cells. Now, this is not absolute. It's not an all-or-none don't eat me signal, but it certainly reduces the uptake of the vector by antigen-presenting cells, resulting in increased efficacy. And I just wanted to share with you some of the data that they published in pigtail macaques with factor 9, resulting in more than 100% factor 9 activity. And this is a busy slide. I just want you to focus on the yellow colored symbols here. These are the animals that received the lenti with the don't eat me signal. These animals have higher levels of factor 9 compared to the animals that had the vector without the don't eat me signal. So there's some logic here that confirms the preclinical results in mice. Now, great integration can give lifelong expression, but it's also a double-edged sword. It may increase the risk of insertional oncogenesis, at least theoretically. We can mitigate this risk by keeping the vector copy number low. Nobody is saying that we should do gene therapy with 10, 20, 30, 40 copies of the gene of interest per cell. We're not going there, when we should never go there. But even within a safe context of 1 to 2 copies per cell, we have to, of course, try to safeguard, try to ensure that the safety is as high as possible. And the devil is really in the details here. And it all boils down to how you design the vector. And there's some elegant work done also by Alessio Cantore from Luigi Naldini's group and collaborators, Eugenio Montini and others, that show that the positioning of the regulatory elements that are needed to drive expression of factor 9 is critically important. So in case you have a strong enhancer and promoter embedded within the lentiviral vector itself, so not in the LTRs, but in the vector, then the cancer risk is very low indeed. They, just like in our experiments that I reported earlier, they also checked the safety of these integrating lenti in tumor-prone mouse models. They are highly sensitive to developing tumors. And you can see here that these self-inactivating lentivectors expressing factor 9, the SEN-ET here shown in the yellow bar, showed no increase in liver cancer incidence compared to the control, which was injected with no vector at all. But as I said, the devil is in the detail. If you take these regulatory elements and include them in the in the LTR of the vector, then you are bound for trouble, because in that case, you end up with a high incidence of liver cancer. So truly, the design of the vector is critically important here. But it's very reassuring that if your promoter enhancer is within the expression cassette and not in the LTR, you can improve the safety to a very significant extent. So what we know is happening is that lentivectors will integrate randomly, quasi-randomly, in fact, with a bias for genes, and they can turn on oncogenes. We know that this is the mechanism of action of insertional oncogenesis in the context of the leukemia that has been reported in the retro trials, using hematopoietic stem cells as targets. But here it's the same principle, but you can minimize this risk by eliminating this enhancer promoter to the extent that even if the vector were to integrate near an oncogene, it's not going to be able to activate its expression, and it therefore does not significantly increase the risk of insertional oncogenesis. Now, it's all a matter of risk, and risk is never black and white. It's always a relative situation. So how could we further control the integration? How can we further determine where our transgene is integrated? Or how can we specifically modify the factor 9 or factor 8 genes themselves? This brings me to the last part of my talk, where we actually explored CRISPR technology to target, in this case, the factor 9 gene. And we and others have shown that with CRISPR, you can target factor 9 and factor 8 genes. You can also correct the bleeding diathesis in hemophilic animals. And there are now clinical trials that are ongoing using CRISPR technology. But I wanted to draw your attention to some other aspects of the CRISPR technology, which remain somewhat underexposed. So using a dual AAV delivery system, we delivered AAV expressing Cas9 to wild-type mouse and another AAV vector expressing the guide RNA to wild-type mice in order to target factor 9. If it targets factor 9, then we should turn a wild-type mouse into a hemophilic mouse. And that's indeed what happens. So we target exon 1 and exon 6 of the mouse factor 9 gene by co-delivery of these vectors into newborn pups. So remember, these newborn pups, they have rapidly proliferating livers. And this is an ideal hit and run scenario, because using AAV, since it's not an integrating vector, the vector genomes would get diluted. They would be lost over time. So it's a typical hit and run phenomenon. So we can document this by measuring the number of Cas9 genomes, which are transient. This is at day 9 and day 90 post-vector administration in newborn, and also Cas9 messenger RNA expression. And that's actually great, because you don't need lifelong expression of Cas9. In fact, you don't want lifelong expression of Cas9. You just want it to be there for the time that it is needed to achieve the targeted genomic modification of your gene of interest, in our case, factor 9. We didn't detect anti-Cas9 antibodies, no sign of liver toxicity, no sign of liver inflammation, and no detectable off-target effects, but I will come back to that in a second. We compared different guides, short guides, long guides. Some studies claimed that the truncated guides, the 1719 nucleotide guide, were more specific. We saw that they were at least as robust as the 20 nucleotide guides. These are the levels of activity that we plotted here compared to the scrambled controls or PBS controls. And we have a 50% drop in factor 9 activity. So this shows that we had successfully targeted factor 9. We have molecular evidence to back this up, 60% almost of specific targeting in exon 1. We used a number of, let's say, primitive algorithms for off-target effects. We didn't detect any in the 3 highest ranked potential off-target effects, but this was, of course, not very comprehensive. We actually did a whole genome sequencing, and to make a long story short, we used a 100x sequencing platform, and we didn't have any strong evidence of target effects. We may have detected one potential off-target effect here, resulting in a CCA insertion, but even that is not 100% sure that it is actually a bona fide off-target effects. Whole genome sequencing is not the most sensitive way to actually pick up off-target effects, even with 100x sequencing, so deep coverage. But we used another method, SFTCG LMPCR, that has a high sensitivity. And I will try to outline what we have discovered here. Well, when we did the whole genome sequencing, we compared an untreated wild-type mouse genome with the treated mouse genomes, and we did detect something quite remarkable, is that we did see something happening at the level of the factor 9 locus itself. There was actually an insertion of the AAV vector at the factor 9 locus, as you can see here with the circus plot, or perhaps even more clearly so, in this high-resolution circus plot. Every single dot here that you see in this circle is an AAV integration. Green is the factor 9 single guide RNA cohort. Red are the scrambled guide RNA controls. You can see this line here in the X chromosome that actually targets the factor 9 locus. And this is happening in all of the mice that were treated at exactly the same location. So let's zoom into this. And this is indeed the site that has been cut by the CRISPR system. It's almost with nanomolecular precision exactly where you expect it to be. Just a few nucleotides upstream of the protospacer adjacent motif did we see an AAV integration happening. Just to show this in another way with the rainbow plots, these are the experimental groups. These are the common integration sites. We see hotspots of integration in the factor 9 locus here when the factor 9 guide RNA is used, but we don't see that with our control, the scrambled guide RNA. There's no integration in the factor 9 locus. And in fact, apart from the factor 9 locus here, there's in all the other mice from the experimental cohorts, all the other integrations are different. So clearly, it is there's a preferential integration of AAV in this site. So what is happening here is that the CRISPR-Cas system results in a double-strand DNA break in the factor 9 locus, and then this creates an opportunity for the genome to capture DNA that's floating around in the nucleus, like AAV genomes, that then get trapped possibly through a non-homologous end-joining mediated capture in this double-strand break. So we believe that this is what we are actually detecting here using this SEPTS LNPCR methods. At the same token, it got us thinking, and we said, well, maybe we can use this method to also detect off-target effects, since we had difficulties using whole genome sequencing to detect off-targets with high sensitivity. But this kind of approach would allow us to do that. So this has important implications to use SFTCG LNPCR to identify and map CRISPR-induced double-strand breaks, not only at the on-target site, as shown here, but perhaps also at potential off-target sites. So the idea is the same, because from the point of view of the cell, it makes no difference if it is an off-target or an on-target site. The minute you have a double-strand break, it will create an opportunity for AAV to be captured into that site. Now, interestingly, we did, of course, see many AAV integrations, and none of these AAV integrations were near a putative off-target sites. We had a computational algorithm. We tested 5,000 computationally predicted CRISPR off-target sites, and none of the AAV integrations was in proximity of any of these sites. So this is very comprehensive indeed. I mean, you can see on this table here, the closest is about, let's say, the median is about more than 100,000 nucleotides. So it's just a canonical AAV integration, completely independent of CRISPR. And this brings me to some of my last slide. In summary, so we have basically shown that transient expression of Cas9 and careful guide RNA design gives us a platform to target factor 9, perhaps also factor 8, as a hit and run approach for gene editing. And we didn't detect any off-target effects, maybe one, but I'm not entirely convinced. And even with the SFTCG LNPCR method, which is very sensitive indeed, we didn't see any evidence of off-targets. So that's very reassuring indeed. But it also carries a message of caution. You know, there's other LNP-based CRISPR approaches that are being explored, but even in those approaches, donor DNA is supplied for homologous targeting, and one has to bear in mind that these AAV vectors can then also integrate in the CRISPR-induced target site. So we have learned a lot over the past decades, and the knowledge is expanding exponentially. There's a lot that we know. There's also, we also know that we know a lot of things that we don't know. And I think we have to be humble here, that real knowledge is to know the extent of one's ignorance. And I'm a bit philosophical here at the end, quoting Confucius, but I think that's the only way forward. So I think the more we know, the more we realize there's still so much to learn. And certainly hemophilia has been a trailblazer for the field at large. I'm very happy that it's now going to make a difference, in a real difference in patients' life, to treat them by gene therapy. I've had the pleasure to talk to several patients that have been treated by gene therapy, several hemophilia patients. Their lives, at least the patients that I have talked to, has changed drastically. They are off the hook, so to speak, and they can they basically have a life before and after gene therapy that is significantly different indeed. Last but not least, the most important slide of this presentation is to thank all the folks who've contributed to some of the work that I have presented. I've tried to also take the opportunity to give a broader overview of the fields, citing some of the work of other groups. And I'm also very grateful to some of the collaborators that I have mentioned along the way, as well as the different funding agencies that made this work possible. And if you would like to refresh your knowledge on hemophilia gene therapy, I would encourage you to read some of my recent reviews on the subject matter, Hemophilia Gene Therapy, The End of the Beginning. And don't forget to send us your questions via the YouTube live chat. Thank you very much.
Thank you very much, Thierry. Yeah, super. So it was really a very inspiring and beautiful overview of all the work that's been done on hemophilia focusing a little bit more on yours, but nevertheless, we got an overview of everything. I got a couple of questions, and I'm going to end with an almost philosophical question too. You will see. But I wondered if you, with this computational analysis, did you try to find specific promoters for other tissues already?
That's a very important question, Els. Thank you for asking me that question. Yes, it is indeed a platform that we can use to identify cis-regulatory modules that would confer high expression and high tissue specificity beyond hepatocytes. We've actually validated it in the muscle, the skeletal muscle, as well as in the heart, and most recently also in endothelial cells. So we have a whole bunch of target cells where we have identified these cis-regulatory modules that confer high expression. In fact, in the context of the muscle, we've tested it in the context of a Duchenne muscular dystrophy model, where we incorporated that element in a vector expressing microdystrophin, and it resulted in normalization of muscle function after gene therapy.
And in this case, the specific promoter you identified for the hepatocytes, they really don't transduce the Kupffer cells, or they don't express in the Kupffer cells in this case, I guess?
That is correct. That is correct.
And what I noticed, I mean, you didn't extend on that too much, but once I saw as a delivery vector AV9, once AV5, are those the serotypes that are the best for transducing the liver? Do they have a certain tropism for that?
Yes, thank you for that question. And yes, there's some historical reasons why in some studies AV5 was used, in others AV6 or AV8. We typically used AV8 or AV9 or derivatives thereof for our liver-directed gene therapy preclinical studies. In the clinical trials, for example, in BioMarin's Hemophilia A clinical trial, it was AV5 that was used. AAV5 is a bit of an oddball, if I may say, and I don't mean that disrespectfully, but from an evolutionary perspective, if you compare the AAV5 genome with the other AAVs, it's a little bit off. And that may actually be an advantage from an immunological perspective, that perhaps one is less likely to get some cross immune reactions between different AAV serotypes if you have an AAV that is more distantly related from an evolutionary perspective. So yeah, AAV5 was used in the BioMarin trial. Another AAV capsid variant, which was obtained by directed evolution, was used in the Hemophilia B trial from Spark. That was a capsid that was developed initially by Marquet, if my understanding is correct. So yes, there's quite a bit of diversity still in there.
But does this mean that one also could think about injecting another serotype a second time, or would there be too much cross reaction, or is there really an opportunity there? If people really lose the expression over time after 5 to 10 years, do you think it would be thinkable to re-inject?
My understanding is that even though the different AAV serotypes are genetically distinct, and one can end up with fairly different variants or serotypes, still they are sufficiently similar to still that you will not be able to re-administer the vectors. So in other words, you come in with an AAV5, you will have antibodies that will cross-react against AAV8 or vice versa. However, there are other ways to get around this, and there's some interesting preclinical work that's been done where by footprint engineering, where some antibody binding domains of the AAV capsids are being engineered so that they don't bind the antibodies anymore. that can to some extent reduce the reactivity to antibodies, but it's not a black and white phenomenon. It's merely a relative decrease. It's not all or nothing. There are other strategies we need to explore to be able to treat patients that are seropositive for AAV that have high titers of antibodies, or to ultimately treat patients that need to be retreated with AAV.
And one question about the factor 9. So the discovery of the Padua mutation, of course, made a huge difference in the field. Then you added more mutations, which even augment the efficacy even more. Do you actually know what these extra mutations bring to the protein? Does it have a longer half-life? Is it more stable in the bloodstream, or do you have a more exact idea what they actually bring to the protein?
Yes. Well, in this particular case, we do. For example, the antithrombin inhibition is to some extent relieved. There's increased activity per se. It works on different levels, actually. Also for the Padua itself, there's more and more insights how this actually is working. And I think an important consideration here is, yes, it's great if we can always try to make it more active, so or less susceptible to inhibitory pathways, which is what we did with the CB variants. But ultimately, we also have to be aware of the fact that we shouldn't make it more immunogenic. The more mutations we engineer in the protein, the higher the risk that it may become more immunogenic. That is one concern. And secondly, the more active it gets, well, we shouldn't push the envelope to the extent that we may have an increased risk of thrombosis. Let us not forget that Padua was identified in a thrombophilia patient. So, but we are aware of these potential risk, and we address that in the experimental models. We didn't see a significant increase in immunogenicity, neither with Padua, which has a single point mutation, or with the CB variants. So that's reassuring. And others have conducted even more comprehensive studies in large animal models as well. So that's reassuring. As far as the increased thrombogenicity is concerned, we checked the occurrence of D-dimer. D-dimer is a marker for increased thrombogenicity. We didn't see that. There are other assays we could use to test that. And so far, if you look at the literature as a whole, in the context of Padua, in the context of hemophilia gene therapy with Padua, there's no evidence of increased thrombogenicity. However, you know, nothing in nature is black and white, isn't it? There was one patient in one of the trials with the Padua vector that developed a thrombotic event, but it's really not clear whether you could link that to the use of Padua due to the fact that it's an N of 1, and there are other theoretical considerations that do not necessarily support that it's resulting from the use of Padua.
And I think you briefly mentioned, this is a question of somebody in the chat, of Emmanuel Payan. He asks if you propose to modify also the glycosylation profiles at some point. Are you actively working on that, or can you tell us a little bit more about this?
Well, we in our group, we are not working on that actively. There is the group of Steve Pipe that has done great work historically and also Amit Natwani has also worked on some of the glycosylation pattern. So what they have done is that the B domain, if you look at the factor VIII protein, there's a central B domain, which you can actually delete. All of us use the B domain deleted factor VIII. The advantage is that by deleting the B domain, you also eliminate negative acting sequences in the messenger RNA. So you increase transcription. So that's great. But you prevent the intracellular trafficking of your protein. So what Amit Natwani and colleagues have done is they put a little bit of amino acid residues back into the B domain deleted factor VIII, if you may. They're glycosylated, and the hope is that it would help the trafficking of the protein across the ER and the Golgi. And there's some evidence in preclinical studies that he has higher expression levels with these variants.
And then it's not really a philosophical question. It's more an economical question. You mentioned it also. These therapies, the therapy for hemophilia A, B, and a lot of other gene therapies are extremely expensive. So there are people working on that, trying to find solutions. What do you think, for example, in general, but also for hemophilia B, compared to a lifelong treatment with recombinant clotting factor, which is also quite expensive, I guess? Can you comment on that, Thierry, just from your point of view?
Yes, that's a very important question, of course, and financial considerations are important for society, definitely. If all is said and done and you have a safe and efficacious vector, somebody has to pay for it, right? And depending on the country, you have different systems in place. And my understanding is that if you say, for example, that on average, this type of therapies, whether it's hemophilia or some other therapies, they fall in the same ballpark, let's say 2 million euros. Yeah. The current treatment for hemophilia is 300,000, let's say 300,000 per year per patient, 300,000 euro per year per patient, 2 million euro per year. Hopefully, it's a one-and-done treatment. Let's just assume for the sake of argument, it's a one-and-done treatment. That means after 7 years of conventional therapy, you would be break-even, so to speak. So bearing in mind that these patients have, in the Western countries at least, a normal life expectancy, this would be a huge this will have a huge cost-saving impact, undoubtedly, even though the price, paying a bill of 2 million euro at once is a lot of money, but over the long run, this is actually significant cost saving. There's another thing to consider here is that, as always, you buy the latest iPhone, you're going to spend a lot of money, right? But over time, competition plays, you will have also competing products. And then once the competition plays, you can still get good quality stuff for a cheaper price. And I think this is also going to play in these advanced therapies like hemophilia gene therapy.
I completely agree with you. And I thank you really for answering all the questions so very clearly. And I hope that the audience will agree with me. And thank you again for this very, very nice talk. Thank you, Thierry.
Thank you very much, Els, and enjoy the Tour de France and the Olympics.
I will. Thank you. Bye.